the complexity of microbiome research and the application of standards throughout the workflow. Before we get started, let's meet our panelists. Panelists, can you tell us a little bit about yourself and the area of your research? Scott, would you like to lead the group?
Okay. Hi, everyone. I'm Scott Jackson. I'm the leader of the Complex Microbial Systems group at NIST, which is the National Institute of Standards and Technology. And we focus a lot on microbiome measurement, uncertainty, and the application of standards, reference materials, et cetera, to try to improve measurement assurance.
Everyone, my name is Sam Minot. I'm a microbiologist and bioinformatician at the Fred Hutch Cancer Research Center in Seattle. And I work with a range of researchers here at the Cancer Center to help connect their informatics to their science and get us closer to cancer cures using the microbiome.
Hi, my name is Tasha Santiago. I am currently a microbiome discovery analyst at Diversigen in Houston, Texas. Before joining Diversigen, I was working at the ATCC Center for Translational Microbiology, where I was working in developing mock communities and also several different spiking controls.
Hi, everyone. This is Nick Greenfield. I'm the founder of OneCodex, which is a data analysis platform for microbial genomics. And we're very interested in reproducibility, both from the computational side, thinking about how to do bioinformatics and particularly large-scale data analysis in a reproducible way, as well as have collaborated with ATCC on the design and development of bioinformatic analysis for microbiome standards and controls.
Great, thank you. I would like to start by discussing the reproducibility crisis that has plagued microbiome research for years, and how the use of standards can help us identify and address these issues of reproducibility. So the first question I have, and it's for all the panelists, but are the current microbiome standards adequate regarding the complexity and diversity of organisms? And are they helping researchers meet meaningful conclusions?
I guess I'll take a first stab at that. This is Scott, just to maybe set the stage, as I always say on my soapbox, with regard to microbiome measurements, there's bias in every step of the measurement process, from how you collect and store your sample to how you analyze and interpret your data, and everything in between. And that everything in between is your DNA extraction and your library preparation, your sequencing, bioinformatics, et cetera. So you can't point to any one thing and say, well, this is where bias is introduced, because it's literally introduced at every step. So there are a suite of, I applaud a lot of the commercial companies and others who have developed reference materials, mock communities, et cetera, for the microbiome community. And I think that's a great direction. And I think we just need to keep pushing forward in that direction to help the community. Unfortunately, in this space, what we're measuring itself is very complex. So to make reference materials is very, very difficult, nearly impossible. So to really make reference materials that are ground truth, that represent the true diversity that you find in microbiomes in nature, whether it's the gut microbiome or the soil microbiome, where you might have hundreds or different species and endless number of strains within species. So really building a reference material that is ground truth, that represents what a real microbiome looks like, I think we're pretty far from, and it's certainly something worth striving for.
Scott, I think that's a great way that you put it, taking the topic of standards and really pulling out reference materials as a specific way to instantiate standards. It's something that can be so hard to have an analyte where you know it's going to behave reproducibly, and that you have the same input from experiment to experiment. And I think it's good for everyone to note that this is actually relatively recent. From the point of view of academic researchers, having the ability to order a piece of material that you know contains the same organisms for run after run, it's not the complete answer, but it's certainly, I think, a very valuable thing that researchers are starting to bring online and just make part of the scenery for how to do research.
Mm-hmm.
Yeah, I wanted to add that there's going to be room for improvement for sure. And that's probably one of the reasons why the scientific community and companies like ATCC are developing these mock communities and standards, mimicking the gut, the oral, the skin, and the vaginal microbiomes, we also know that ATCC developed the fungal and the viral communities and the spiking controls, which are very helpful and very unique. And I think moving forward, a lot of these standards are going to continue to evolve as the scientific community thinks is the most appropriate, because what we want at the end of the day is to make microbiome research more comparable.
Yeah,
I don't have that much. Oh, sorry.
No, go ahead. Well, I was going to ask, so Tasha, what do you think makes a true standard? Like, what would be helpful, and how many organisms should be in a standard, and what's the type of diversity that researchers are interested in seeing?
No, I think that really depends on the sample type, because there are sample types, like, for example, an infant stool is not as complex as an adult stool sample, right? So it really depends on the type of sample you're working with. We cannot really compare a stool with a soil microbiome or a water microbiome. So it really is going to depend on the complexity of the sample you're dealing with.
I absolutely agree with that. And I'll just note that I'm in a position where I collaborate with people that work on really different types of microbiomes. And there are differences when you compare the gut versus the skin versus the female reproductive tract. And when you look at the abundance of the most abundant organism, in some samples, you might be looking at 90%, and in others, you might be looking at 9 out of 1,000. Mm-hmm. So I think it's right to say that there is no one answer.
Yeah.
Scott, would you like to add a little bit of information about how difficult it is to develop a microbiome standard?
Well, Brianna, I think you would probably know better than me, since ATCC has developed a catalog of them. I'll just say in a very general way, in order to make a microbiome reference material, aka a mock community, you have to be able to enumerate bacterial cells accurately, and that is not trivial. Yes, for E. coli and maybe Saccharomyces cerevisiae, we can accurately enumerate cells, but not all bacteria look like these perfect rods and spheres. They form chains and clumps and filaments, and sometimes, we see structures under electron microscope that look like cavernous, structures that are, clumps of bacterial cells. So, yes, not all bacteria are easy to grow in the lab. A lot of these are unculturable, so we can really only grow what we can grow. If we can grow them, it doesn't mean we can necessarily count them accurately. So that's from the mock community side. I will say from the other side, one of the things that we're working on at NIST is developing a human gut microbiome reference material that is poop. So NIST is developing standardized poop. It is not ground truth. We don't know the correct answer, but we are making large amounts that we know are homogeneous and stable. And going back to what, something Sam said earlier, just having abundance of material that is homogeneous and stable will allow labs to harmonize their measurements and understand if their measurements are comparable within a lab, to understand reproducibility within your own workflow from day to day. Having a common material that is shared, that is abundant, that is homogeneous and stable, can be very powerful, even though it is not ground truth.
No, that's a very good point, and that actually leads me into a question I have for Nick about bioinformatics and the analysis of any type of microbiome sample. So Scott just alluded to the homogenized samples that he's working with, but depending on the database that data is bounced off of, you could end up with a very different result. So Nick, in your line of work, how do you mitigate that, and how do you control for that in the future to improve the data that's coming off of your whatever type of analysis that you're doing?
Well, I think the key. Oh, sorry, someone coming to the door and the dog in the background. Work from home. I think the key to enumerating kind of all of this accurately is to really know, the nice thing about these standards is that we know what's in them, right? And sometimes we know what's in them fairly well. They're draft, modest quality genomes available. And I think that we can improve kind of the access of the quality of reference information that we have available by doing, gold standard assemblies, Oxford Nanopore and Illumina kind of hybrid assemblies or the like. And I think that, even for something like what Scott is describing, if we have a lot of homogenized stool, at very large volumes. There are other techniques that we could apply to better understand what's really there and to get some additional ground truth, at least via kind of an orthogonal method. So maybe it's not just short read sequencing, but we do, you know, tons of ONT metagenomics, or we do high C, or we do, you know, one of these 10x methods that lets us kind of pull out synthetic long reads and get a better sense for what's there. You know, we've then turned that into the NIST stool metagenome assembly challenge or something. But I think that we can augment, you know, we can still use materials where we don't know exactly what the composition is by using other methods to characterize them more deeply than we might characterize your kind of run-of-the-mill sample. So I think there are a bunch of different possibilities around that.
I agree that once you have a material that's available to the community, that the knowledge about that material will only improve because people will want to explore different methods for that. We've seen that with a human genomic DNA reference material that NIST put out, which was certified for its genome sequence. But once the community got it, they began doing these, as you mentioned, you know, long read technologies and doing better assemblies. And the genome sequence and the assembly of that reference genome is now, you know, of high quality because of the community's input.
If I could just add, I think there's maybe a useful analogy to other types of omics technologies for people that might be new to microbiome and what the technologies are that we use with sequencing. So if you look at mass spectrometry, if you're doing mass spectrometry on stool or blood or something like that, you have, you know, isotopically labeled controls where you know exactly what each molecule is. And that is like the defined communities that we've been discussing, like with ATCC. And then it's also useful in mass spec to do something like mixing your entire batch together and putting a small sample on in between every 5 other samples so that you can tell if your machine is drifting. And you don't know what all the metabolites are on targeted metabolomics, but you know that you should be getting relatively the same answer. And so I think that this idea of having multiple types of standards is something that people have to do across omics technologies, and we can probably draw pretty clear parallels to the microbiome.
I wanted to add, at least when you're benchmarking a bioinformatic tool, we notice here in Diversigen that adding a mock community is very helpful. And this happened to us when we were beta testing the ATCC's viral mock community, where we noticed that a lot of these tools out there for viral research provide a number of false positives and a number of false negatives, which of course you don't want in your results. So I think adding standards in the process of benchmarking any bioinformatic tool is very, very helpful.
Sometimes it can be a little scary, can't it? You run a standard and you say, oh, what have I been seeing that I shouldn't have been seeing? Where have my blind spots been? The first time it can be a little bit hair-raising, but still the right thing to do, and it's better in the long run.
Yes. Yes. We've purchased the commercially available mock communities from various commercial sources and run them at NIST, and it's not uncommon for us to get the wrong answer. And in a way, that doesn't bother me because I know that there's bias in my measurement process. I know that my DNA extraction is not 100% efficient across all cell types. I know there's PCR bias, quantitative bias in the library preparation step when you're doing like a Nextera. I know that my measurements are biased. So in the way I think about it, if I had gotten the right answer, I would have thought, well, that can't be right.
So Sam and Scott, actually, I have a follow-up question for both of you. So with so many studies and so much data in the last several years, maybe we need to look beyond DNA and take a closer look at the metabolome or transcriptome or proteome. Any thoughts?
I think a lot of researchers are turning in that direction. You know, ultimately we want to understand sort of from a functional perspective what these microbiomes are doing. You know, when you have a therapeutic formulation like Series Therapeutics has a concoction of bacteria that's therapeutically efficacious, like mechanistically, physiologically, what is responsible for that observed efficacy? And that's a functional measurement, and DNA-based measurements don't really get you that level of granularity with respect to function, and that's where metabolomics comes in. And for our fecal reference material, we are putting a lot of our mass spec analytical chemistry folks on it to begin to put some some measures on some key clinically relevant metabolites. You know, you hear a lot about short chain fatty acids and things like this. So I believe a lot of researchers who have been doing metagenomics historically are turning towards more functional measurements in order to try to understand better what these microbiomes are doing.
I would definitely agree with that. And I think that the last thing you said of using these tools to try to understand really gets to the heart of what we should be talking about when we talk about reproducibility. You know, there's a superficial reading of the word, which says, I run a study in a population, I observe that bug X is associated with disease Y. If it's reproducible, I can run a similar study in a similar population and observe a similar association. But that doesn't actually tell me what is happening, what the function is. And so I think that being able to go from these surveys to some biological entity that I can actually show in a controlled system. Here's an isolate that has a reproducible effect in a mouse model or as a diagnostic or as a therapeutic. I think having a certified therapeutic with defined contents, that's, you know, we're getting closer to the gold standard of reproducibility when we reach for that goal.
Yeah. And if you think about...
Oh, sorry, Scott.
No, go ahead, Nick.
I was going to say, just to chime in on that and how it ties back to this question of, you know, do we have the perfect standards yet and where do we need to go? I think a proxy that some are using, and it's a very convoluted proxy, right, but is looking at strains. And I think one shortcoming of kind of current standards and something that, you know, might be bear thinking about going forward, or can we actually, are there methods that are better and worse at teasing out, you know, differentially abundant mixtures of strains? That's especially challenging bioinformatically, actually. But I think also it's worth thinking about, you know, in the context of PCR and extraction biases, et cetera, you know, can you disambiguate the case where you have a mix of, you know, 100% all strain A of E. coli and a mix where you have, you know, a bunch of kind of commensals and then a little bit of this extra special strain, which might be a good proxy for, you know, a therapeutic that a Series or a Vedanta Bioscience or one of these live biotherapeutic companies is using.
Mm-hmm.
I didn't mean to cut you off there, Scott.
I actually forgot what I was going to say.
Well, actually, it kind of ties into my next question that I have for Sam. And with the well-known reproducibility challenge, Sam, how do these implications impact personalized medicine? And how are researchers making insightful correlations when we know there's so much bias?
Yeah, absolutely. It's very important, and that's something that I think is particularly highlighted by getting to work at a cancer center, where the goal is really to take all these findings that we have, all these associations, all these experiments, and turn them into something that will make a difference for curing cancer. And that's a pretty lofty goal, and it kind of shines a harsh light on approaches that don't end up yielding something that might be able to be translated into a live biotherapeutic or something that's a bit more actionable like that. And so I think standards are absolutely an essential part of this, where we have collections of strains, where we know the genome sequence, where we have mixtures that are defined at the molecular level so that we're sure that our instruments are behaving properly. I think that the requirements for reproducibility and the fidelity of our science really are raised quite a bit when the end goal is we're going to go into a trial of trying to treat people who have cancer. I'm sorry, I might not have answered your question completely, but I think that this is very much what we've been discussing, and I think this is something that a lot of the researchers that I work with have at top of mind.
I'll add to that. I think one of the most exciting discoveries recently with respect to the gut microbiome is in January of 2018, it was reported in 3 back-to-back papers in Science, all 3 labs essentially with the same finding, and that is the gut microbiome modulates the efficacy of immunotherapies, these checkpoint inhibitors that are used to treat cancer patients. And probably a year after that paper, I was invited to an NCI meeting where a lot of the oncologists who published this work met at NCI to discuss how do they leverage this and take advantage of this. And, they started doing FMTs to try to improve the outcome of a cancer patient before they start this immunotherapy. And they find with some FMTs, they become more efficacious, but others have no effect. So there's this idea that there's super poop out there, but what is the super poop? And you can't define it. And that's a measurement problem.
Yeah, I think one thing that is worth noting about all three of those studies is they all did 16S sequencing too. So, much as the progression from kind of higher level taxonomic groupings like we can pull out with 16S to shotgun sequencing, I think eventually strains or functional markers are going to be a really important part of this picture as well. Mm-hmm. So hopefully we'll see the double click of that study maybe next January.
Mm-hmm.
Yeah, that experience, I think, is a great example of how you do an association study and you see that there's some effect. And then when you ask if it's reproducible, the next step is let's take that stool and put it into mice. Because what we really care about is, can a controlled experiment reproduce the effect? And how much better that would be instead of putting just stool in, if you're able to test, batches of 10 and 20 strains where you know exactly what strain is in the sample. That's a world that I think we're all really hoping that we can get to soon.
Scott, I want to circle back to the earlier conversation where you had mentioned the samples that you have, that the study that you're going to start here in the near future with your homogenized stool samples. How did you go about, what were some of the requirements for your donors? And was there something specific in that microbiome that you sought out, or was this just donors and then you move forward from there?
So we're in our phase 2 pilot studies because any NIST reference material that's developed has to be demonstrated to be homogeneous and stable. And people hear that and they dismiss it immediately. But when you're actually tasked with doing that, it really that's all you think about is homogeneity and stability. If we were producing pure silver as a reference material, probably pretty easy to make something that's homogeneous and stable. But when you have fecal material that are full of live microbes and volatile organic compounds, we want a material that is shelf stable for 5 years. So how do you make something that's so biologically complex stable for 5 years? So we've been experimenting with different stability methods. Obviously, these things take time. We can't speed up the hands of the clock. So, this might take a year to test some new stability methods, whether it's -80 or some, buffer addition to it. That's not really what you asked. What you asked is how did we choose our donors? And while we don't expect for these fecal materials to be representative of any communities, we are providing a suite of materials that came from different cohorts of donors. And the only really requirement was that we wanted donors that would have drastically different contents in their fecal material, because if you buy a reference material and there's 5 tubes of poop in there, you'd want them to be different so you can compare things. So right now in our phase 2 study, we just collected fecal material from a cohort of vegans and omnivores. And because diet is such a major factor that influences the content of your gut microbiome, we presumed that these would be the most different in terms of their microbial content and their small molecule metabolite contents. As I think Sam mentioned earlier, to put a certified value on a metabolite, it requires you to have a pure standard, maybe an isotopic analog of that molecule, something you buy from Sigma Aldrich, in a highly purified form. And that's difficult to do, but with this new model that NIST is sort of implementing when it comes to metabolomics, we can provide pseudo-quantitative measures of various metabolites. So if we have a suite of materials, we can say that glucose, for example, we don't know the absolute concentration of glucose in any of them, but I know sample 2 has twice as much glucose as sample 1, and sample 3 has a third as sample 2. So I call this pseudo-quantitation of metabolites. And you know, that's sort of the approach that we're taking now.
Okay, thank you.
Mm-hmm.
Tasha, my next question is for you. In your recent work with the virome, can you allude to some of the challenges that you experienced during that research and how some of those challenges maybe differed from your earlier microbiome work?
So I think we have encountered two major challenges. I think one of them is the sequencing depth, and that's a question that we don't tend to think about, but we have clients that send their samples and then they ask, so how deep do I have to sequence my samples to have good information? And the second challenge is a bioinformatic analysis tool that I think a lot of people have challenges with. So in Diversigen, when we were beta testing the virome community, we tried to sequence it as deep as possible. And as I mentioned very briefly, we have benchmarked and tested a number of different virome annotation tools. And when we were beta testing the virome community, we noticed that, for example, Kraken gives a number of false positives, and we couldn't find some of the viruses that were supposed to be in the mock community when using a number of different bioinformatic tools. In terms of the sequencing depth, I also wanted to mention that we did a very quick experiment here in Diversigen where we sequenced stool samples to 5 gigs of data, which is probably 70 million reads, which is not super deep, but it gives you enough information. And we in silico subsampled the reads down to 0.1 gigs of data, which is probably about 0.3 million reads. And we noticed that the organisms in the stool sample that were present in very low relative abundances were no longer detected at probably 2 to 1 gigs of data, which is about 3 to 4 million reads. And I forgot to mention that we use Metaflan to annotate the samples, which is a marker-based tool. And I think the cool thing about this is that we added ATCC's MSA 2002 as a control. And I really like this mock community because it has organisms in staggered concentrations. And I think to some extent that mock community mimics the stool sample because it has organisms present in very high abundances and very low relative abundances. And we noticed that at 2 to 1 gigs of data, we were not able to detect the organisms in the mock community, which is similar to the stool sample. So I think the mock community, at least in this case, when you see MSA 2002 or even any other mock community, some of them can really reflect your sample of interest. And I think sometimes they can be used not only to control and standardize your pipeline, but also probably to predict some of the outcomes.
I'm interested to hear about a viral standard. I used to do some virome work, and the challenges of bacteria are just multiplied many fold over.
Yeah, I know. I was involved in virome research and I understand it starts from the DNA extraction and then do you amplify the nucleic acids, and it's more challenging, I know.
I can tell you there are a lot of people thinking about viral RNA standards right now.
Yeah.
So Tasha, did you have a preference for going, like with the RNA viruses, did you do a cDNA step or did you do an RNA library prep? And did you notice any difference?
We did the cDNA step.
On the topic of RNA, actually, I'd say that RNA is something that I'd really love to see more standardized protocols for. I think that sequencing even bacterial RNA is quite a bit of a challenge now, specifically thinking about extraction methodologies that are relatively representative, also being able to deplete ribosomes. There's so much ribosome, there's so much host RNA, so the challenges of viruses multiplied by the challenges of RNA, I'd say RNA viruses are something that for microbiome technologies, it's really hard right now.
Mm-hmm. You all probably heard this COVID-19 is being shed into the fecal material, into the feces of the infected patients, and this presumably is unusual for upper respiratory virus. And that opens up some unique opportunities and challenges. Number one, fecal material could be a new diagnostic target to test people for the infection. But also, and the FDA has already released a guidance / warning about this, that FMTs that are being used widely across the country for various clinical trials, they're going to have to now be screened for COVID-19, which means you're going to have to do these RNA-based nucleic acid tests on fecal material. And that's going to become a real common thing. So, yeah.
I know one of the challenges that we had during the development of the Bioinformatics standard was just the sheer number of host DNA reads, because all the different viruses within that virome, they all have a different host cell. So Tasha, can you speak to some of the challenges that you experienced during the bioinformatics analysis and separating the host reads from from the viruses that you were trying to discover?
That's a very good question. I think the removal of the host sequences, it's a must when you do viral research. It's usually done after you sequence, right? It can be also done, I guess, upstream. But it's a challenge for sure. But I think it also gives you the opportunity to, for example, and I mentioned this to the rest of the team, to test different methods to remove host DNA. It gives you the opportunity, because I noticed this mock community, you mentioned that it has a lot of host DNA. So I think it gives you the opportunity to test different depletion methods.
Exactly, and it would actually mimic a sample, as Scott was just referring to, that when you get these virus samples or these clinical samples, they come with the host DNA.
Exactly, yeah, exactly.
So Nick, my last question is, how reliable are bioinformatic tools that are being developed for the use of clinical or the translational level? And how is One Codex addressing these challenges?
Reliable. I think that there's a lot of things to unpack. So, I mean, I think taking a step back and kind of looking at bioinformatics software development through a broader lens, I mean, I think all of us have experienced the challenge of, there's a paper with an interesting series of results. There are scripts available upon request for the analysis, right? So sometimes I think reproducibility challenges start simply from lack of access to the data protocols being used as kind of first-class analogs to the laboratory protocols that are being published. Sometimes code is available, but it's difficult to run or almost impossible to run. And I think increasingly, we're seeing better and better kind of software practices, better and better workflow management tools, containerization strategies, et cetera, that are helping kind of like raise the starting point from a reproducibility point of view of tools coming out of the academic literature. But I think, tools that researchers are developing for specific publications tend to be, designed to have that one novel feature to them, or to help achieve and uncover specific findings, right? And those tend not to be general purpose tools. They tend to be the types of tools that there's going to be less investment in testing and maintenance. And so I think, there's this huge tension between the new cutting edge tools, which may be the best from some of the best thinking and most novel ideas are concentrated in these kind of cutting edge research tools, but there's a tension because the incentives for those tools are often not, software that works reliably on many platforms and is easy to package and encapsulate and is well maintained and tested and is tested continuously, et cetera. So I think that's kind of at the broad level, a bunch of tensions. I think things are moving in a good but slow direction. But I think researchers are increasingly adopting tools like Nextflow and Docker or Singularity containers, et cetera, and those things make a big difference. In terms of translational work and what companies are doing, I think it ranges from folks who are following kind of ISO software development standards, which are good guidelines, but don't, they're simply guidelines. They tell you one way you could write good software that's reproducible, but they don't enforce that the software actually is reproducible, right? And so I think, there are all these tensions between, we have some guidelines that if we follow them, they help, but they don't kind of enforce good outcomes. So I think, what's really needed in this space, and there are more and more companies doing this, but are organizations that are focused on, software development of bioinformatic solutions as an engineering discipline, which means rigorous testing, which means things like continuous integration, so testing continuously across known and unknown data inputs, et cetera, to kind of raise the level so that we can believe when we're doing research, and then we're doing something in a translational context, and then finally we're moving it into a clinical context, we're using the same tools rather than using a different tool at each step, which has its own biases and problems. And we believe the tools perform the same. The clinical, execution of the tool shouldn't really be different from the research execution of the tool. But I think often that is the case right now, because different groups are responsible and different stakeholders are involved, and things get rewritten and repackaged throughout that process. And that's a source of, a lot of, probably some very minor issues, and then some kind of unknowns that could be significant. But yeah, I think so those are some of the tensions. And then I think there are a lot more in terms of keeping things up to date. If you talk to folks in clinical genomics on the human side, you'll hear stories of people using versions of software that are 3 or 4 years old that have known bugs in it, because that's how they did their validation. And the process of revalidating isn't clear. It's not easy. It's not push button. So to upgrade your aligner from BWA, I don't know the BWA versions offhand, but like 1.7 to 1.10, let's say, which is known to improve things, is something that a lot of groups aren't doing. And so I think there's a whole infrastructure problem that this community hasn't really figured out, and it's really hard to think about how do we do validation, how do we make it easier to do revalidations of databases, of new versions of software, et cetera, so we don't kind of keep all this arcane, outdated software for 10 years as our solution to the problem, because that's not a great solution either.
Yeah. I want to add one more piece to the bioinformatic analysis. And that is, when you do shotgun sequencing of a human gut microbiome, it's not uncommon for 50, 40, 30% of those reads to be unmappable. That is, there's nothing in GenBank that they map to. These are reads from organisms that we have no reference genome sequences for. So it sort of begs the question, well, how representative are our databases? And it's something that me and a colleague kind of sat down on the back of the envelope and we're asking ourselves one day. And we found a PNAS paper that was published in 2012, and the investigators set out to determine the number of species that exist on Earth, right? So they used a lot of metagenomic data from the Earth Microbiome Project and did some modeling. Long story short, the number they came up with is 10 to the 12th. They determined that there are approximately 10 to the 12th species of microbes that exist on Earth, right? So that's sort of the denominator. Now, what's the numerator? The numerator is the number of species that exist in our database. So you go to GenBank and you ask, well, how many species are represented? And it's about 25 to 30,000, right? So that's the numerator. So 30,000 over 10 to the 12th is like 0.00001%, right? So, we've got a long way to go with respect to populating the reference databases in order to be able to see what we're really trying to see.
Yeah. Though it's not that bad for the gut microbiome. It's not like 0.001% of all the reads we can classify. But no, I think that's an absolutely fair point. And, just to chime in on that, like, we know we have a database from right now. In 2 years, we'll have better information about what's in a gut microbiome sample. It's important informatically that we have the ability to say, let's do the same analysis with a new database, and let's understand what those differences are. And let's make sure that we're comparing apples to apples with our results and what we're reporting, et cetera. This is, it's kind of analogous to switching your variable regions in a 16S study and expecting it to get the same result. We're not going to switch databases and get the same result. We're going to get a different lens into the truth. And we need to make sure that, we have the methods and the infrastructure to accommodate doing those types of updates and getting those, refreshed perspectives.
Mm-hmm.
Yeah, I think sometimes it can be hard to convey to people why it's so important to have a system that's continuously tested as you change the database. Because if you're new to the microbiome, you might say, we'll just make a good database. And if you have to update it, that means that there was something wrong before and you fix it. And no, that's not true. We exist on a moving frontier of discovery, and every month people are discovering new species in the gut that we never knew about before. And that's not a failure, that's a success. And so we need to have systems where we can analyze, knowing that moving forward, we're maintaining backwards compatibility, but we're still leveraging this new information. And we're recognizing that there's going to be more information next year and the year after that, which is, it's hard to be comfortable with uncertainty. I think that might be the motto of this year, but that's something that I think standards will definitely help us with.
Yeah, and metagenomics is not a multiplex panel, right? And I think sometimes people talk about metagenomics, particularly in an infectious disease context, where the analogy is a little bit better, as kind of like megaplexed PCR equivalent. And, you know, that's partially true if you know what your targets are and you only look at those. But when we look at microbiome samples, that's not how we use the method at all. And so to Sam's point, I think it's absolutely true that there is a moving frontier of knowledge here, you know, and we need to balance having our data analysis be comparable with making sure that we're as close to that frontier as is reasonable, you know, for a given study and a given application.
Mm-hmm. Something that came up in our workshop, and this is building on exactly what you're saying, Nick, that if you have a method, we talk about these microbiome measurements being biased and there's so much measurement uncertainty. Well, how is it that these, you know, Science and Nature papers are getting published every other week? And the saving grace of these measurements is that they're very reproducible if you use the same method. And what that means in a laboratory that's looking at two cohorts, if they see differences, those differences are probably real. Now, any single measurement, any absolute measurement is not going to be correct. That is, they're not accurate, but rather they're precise. So, you know, that's, say it another way, everything is relative. You know, it's if you see differences between two cohorts of disease versus healthy, and you see differences in relative abundances, these are probably real if the method used to measure them was a lockdown standardized method, no matter what it is, no matter what. And so sure, you know, changing your software version, changing your DNA extraction method, you're going to just now make your data incompatible with what you previously have generated.
And this is where putting good controls in alongside all your samples, you can then get one measurement and to the question of, you know, is this study, this cohort is the DNA being extracted or the libraries being prepped, et cetera, with similar biases to the biases in the other study. And that's not perfect. And analytically, we don't have all the tools to do that. But it provides some handrails at the very least and a little bit of comfort. And then I think it's a foundation for hopefully some better methods going forward.
Mm-hmm.
So thank you. And just to follow up with what Sam said, this is the time to be uncomfortable with uncertainty. And he's absolutely right. And that's where we're going to see the greatest growth. We do talk about reproducibility issues and the challenges, but we are going in the right direction. And I think this has been a very insightful conversation. So with that, that's all the time we have for today. So I want to thank our panelists. This was very enlightening. And thank you to the audience for joining us today.